enzymatic digestion, and immune response. So the naive way to design antisense oligonucleotide analogs is to only consider the two-state model of probe hybridization, which describes the probe and its hybridization to its complement. Target accessibility and tail folding are not accounted for in this model, and this is the most prevalent model in any commercial software that we found outside of our own walls. And so there's an incomplete model of what is really happening in the system, which leads to a lack of sensitivity due to target and probe secondary structure. So the platform for each of our DNA software products is based on this multi-state equilibrium model, and this accounts for the energetic cost of targets and probe secondary structure through coupled equilibria. This means that it'll ultimately increase assay sensitivity and reduce false negative results, as a target or desired target may be involved in secondary structure, and it takes an energetic penalty to break that apart so that you can access that target. And this is the sort of equilibrium that we model in all of our products. So this means that Antisense Architect will evaluate and thermodynamically score your desired design sites. And if given an entire target, it will identify all thermodynamically accessible targets that the user specifies under their own conditions. So this will give you the most sensitive oligonucleotide designs. So the second way that we solve the problems in the antisense design industry is that we use thermoblast. So many researchers use NCBI blast, and they sometimes confuse it with thermoblast. But through NCBI blast, when they query their oligonucleotide designs, typically the #1 answer we get for that use is because it's free. But even free has its associated costs, and blast is the wrong tool for determining selectivity. So blast was meant to determine common evolutionary ancestry through sequence similarity. So you can imagine, as everybody's done, you take the reverse complement and then you just blast that against the sequence. However, sequence similarity does not equal thermodynamic stability, as blast does not account for thermodynamic contributions to base pairing, mismatches, target bulges, or dangling ends, in addition to the different oligo backbone, sugar, and base chemistries that are available from DNA software. So DNA software's thermoblast takes all of the aforementioned thermodynamic contributions into account in the design of the antisense oligonucleotide analogs by scanning design candidates against entire messenger RNA libraries, which minimizes off-target effects to address the side effects associated with undesired gene silencing. So it's well documented in the literature that the key to preventing enzymatic digestion of antisense oligonucleotides, or in some cases even immune stimulation, that lays in the chemical substitution of the probe backbone, the 2' sugar moieties, or even the bases. So what's not well documented in the literature are DNA software's proprietary thermodynamic databases that are required for the rational design of antisense probes that take into account the thermodynamics required to predict maximum knockdown efficiency. No other software products on the market have the required chemical modification packages available that can predict modified probes, particularly their secondary structure or the energy of hybridization, and ultimately the melting temperature, or TM, which many people focus on as an accurate antisense design. Only DNA software has the capability to build an Antisense Architect platform that is specific to your research needs and requirements that reduce or negate the enzymatic digestion of antisense probes and the solicitation of the immune response. Okay, so then what more directly about Antisense Architect? This is a summary of what I've just discussed. And it really addresses all of the recurring issues of target accessibility, probe design, off-target effects, and the proper use of chemical modifications that are thwarting the antisense industry today. So each of these problems are solved in Antisense Architect, which allows researchers to rationally design antisense oligonucleotide analogs using proper design principles that are engineered to modify the expression of targeted genes through optimal knockdown efficiency. So many of these design heuristics that you see on this slide will be discussed in the software demo. So in the demo, I'm going to show you the actual Antisense Architect, the application, current application that we have on our cloud-based portal. So we've chosen a cloud-based portal because it's able to move a little faster as we have more computational capacity, and it's also more accessible to any researcher from around the world. So it's a wizard-based design, which means that any people who are familiar with our product VisualAmp know that it's a power tool, and there's a lot of background knowledge that goes into using that tool. However, with the wizard design, we boiled it down to 4 easy steps for Antisense Architect designs. And step 1 is to choose the background ref-seq of interest and the target within that ref-seq. Then you're going to choose a desired probe chemistry. Actually, there's 2 steps. And then the 3rd step is to choose the desired probe chemistry and hybridization conditions. And then you just run the job. So the example that I'm going to show in the demo is actually based on this article here. And if you would like this information, then you could contact us after the webinar, and we could forward this to you if you'd like to work with our Antisense Architect using this literature. So I'm switching over now to the DNA software application portal. So again, this is a complete portal. We have a pull-down menu, and as you see, we have many different products in our portal now. But we're going to focus on Antisense Architect. So once you've signed in, we can go to a new project. So within the new project, there's going to be a description on what is happening and what you're doing. We also have a box that can show tips. So instead of having a reference manual, we have something that's already loaded behind the software. So as you move about, it'll send a window up for you. So what we're going to do is search a playlist depository that we have, and we've amassed one of the largest playlist depositories out there, and particularly for antisense studies, a very important model is a zebrafish. So we pull up the zebrafish. So we've completed step 1. We're going to select our background. And once you select next, you see that we have all of the different sequences. And these are listed. These are already pre-formatted. These are all from NCBI. And you're able to look at those sequences more closely through an NCBI link. And you could also even download these sequences. But what we're going to do next is we're going to select a target. And to do that, we're going to use the accession number. So I'm going to input the accession number, or if you know the common name, you can input that information in this search box. But we're just going to go directly to our target. And this is the target that they actually used in the literature reference that I presented. And we're going to try and knock down a transcription factor in zebrafish. So we're just going to select that as a sequence. Then you select next. So now we've completed step 2. So the next window is going to show you the type of format. And again, this is adaptable to any application that you're using. For this particular webinar, we're focusing on Antisense. We have a variety of backbone chemistries that are already on this platform. We're going to go with Morpholino. You can choose a length. So we can go from 20 to 30 to give us a wide variety of different designs. A lot of people like to focus on a single length. In that case, you would just input that length for both the minimum and maximum. Select next. So we've defined our chemistry, and now we need to define our range. And so what I'm going to try and do is, this is the actual knockdown that occurred, or how we modified the gene transcription in the zebrafish. And so you notice we get this picture in our view. We can hover and look at all of the different landmarks that are in the gene of interest. And what I want to do is, what's shown here, is I want to put a blocking Morpholino probe right on the 3' end of this, or the 5' end of this, I'm sorry, so it's messenger RNA. And so in this way, we're going to be able to modify the genetic expression of this transcriptional repressor protein. And we notice at that position that we could also zoom in if we want to get a better idea of where we're at. And it's a click and drag mechanism. But we're going to select that one position. And we're also going to then define the tail folding. So within the tail folding itself, we can use large tails, but quite often people are using smaller tails. So for this particular example, I'm just going to select tail folding of 50. So what this is going to do is it's going to fold those tails, 50 nucleotides, 5' and 3' of the target site, and it's going to evaluate how accessible the defined target is in these ranges that we've already defined. So then we just select next. These are the hybridization conditions. For this particular demo, we're just going to leave the concentrations. We also have a drop-down menu that has many of the most popular packages out there for enzymes and salt. So we're just going to choose typical endpoint PCR. And you could also define a new kit. So if you have a proprietary kit that you like using, one that you use just in your organization, you can define that right here. Give it a name, input all of the concentrations, and save it. And then it will populate to this drop-down menu. But as you select the menu, it will also reaffirm that this is the correct choice, as all of the concentrations will pop up in this window here. So then we just select next. So to finish with the chemistry, a lot of people like to use 5' dyes, sometimes 3', depends on what you want to use. We do have another drop-down menu that has all of the most studied fluorophores in here already. So everything that's already in here, the thermodynamic parameters have been determined in my lab, and we've been able to input those into our platform. So for this particular one, we're just going to choose a FAM. We add that. So we're going to ask that we have a modification that has a FAM so that we can find it in our assay. So we select next. And here we have a lot of the different heuristics that are used. Many of the default heuristics work very well. However, there is a chance for advanced users to really start to manipulate their target and their target folding. And many of the heuristics that are well known to most people are target accessibility. So this is where it's looking at secondary structure. Probe complexity, in that we don't want to have runs of A's, C's, T's, or G's, or any repeats like GT over, or GU over again. And it'll do the same thing for the target with all of the poly metrics here. We could look for purine runs, weight oligo length, and even CG steps if we want to start to consider some of the immune responses that might occur. We can select how many candidates to return. So if we only want to order 10, we can just input that. And we're going to calculate the specificity using ThermoBlast, which is also on the cloud portal. So we've completed step 3 by assigning all of our conditions and chemistries. So then you just select run. So you're going to see a little cloud wheel pop up like this, letting you know that everything's running. So it's now sending it to the cloud. And what's going to happen is you're going to wind up getting all of the Antisense Architect jobs that you've run previously. So you are able to retain all of the information that you've input before and all of the results that you've had before. So under the results, this is one that I ran earlier today, and while this is cooking, and it does take about 5 minutes for this to work because it is running through an entire mRNA background. So I'm just going to open up the previous results that I had earlier. Okay, and this is what the results are going to look like. So I asked for 10 probes, and it gave me 10 probes. So here are all of the different heuristics that are used to rank the probes, and they are all ranked and given a total score based upon the heuristics that were assigned. You could either use the default, which worked very well, or you could use your own heuristics. If there's something unusual about your target and you're very familiar with that, you could also assign that here. And so if we look at the results, we see that we have the actual result from the literature, which showed up here. And this was shown in the literature to be very effective in knocking down this transcriptional repressor protein. But what was interesting about it was Antisense Architect actually found another probe that worked even better than the one in the literature. And so we look at the differences here, and one of the major differences was that we have the difference in our metric here for CG steps. So within the CG steps, we have a penalty of 0.75 because CG steps in the correct sequence motif can elicit an immune response and recruit antibodies. And each one of these has 3 CG steps. There's one, and there's the third one. So they both have a penalty of 0.75. But you'll notice if you look at this CG step, there are 2 purines, immediately 5' to that, and 2 pyrimidines, 3' to that. And so for the one that was used in the literature, this one would actually elicit an immune response if it were used in other systems. I'm not sure if it would do that or what the biology is in a zebrafish, but it's something just to be aware of if you're applying this also to human subjects or human models. And some of the other metrics in scoring is we see that we have the nucleotide position, where this thing is located on a target. The sensitivity is essentially the amount of the target that is taken up by that probe under the experimental conditions that the user defined. Specificity is the ThermoBlast run, and the ThermoBlast run will finish in typically 15-20 minutes. And that will give you specificity, and sometimes, depending on the specificity, it's going to come back and re-rank those based upon any off-target scoring that it gets. So it might re-rank your final probe designs. So target accessibility is looking at the secondary structure of the target. Probe folding is ensuring that you don't have the any hairpin formation that would interfere or outcompete binding or bimolecular binding to the target. And then there's a complexity penalty. So within the complexity, we're looking at things like this. So we did get a little bit of a penalty here for this because we do have some runs of purines here. So we have a large run of purine, and it's looking for complexities that are 3 nucleotides or larger than 3 nucleotides. So it won't be penalized as much for having 3 cytidines in a row, but it is being penalized for having 6 or by having all of these purines in its composition. So we also have any of the polys. So again, it triggers for anything that has more than 3 nucleotides in a row. None of these have that, so the penalty is all 0. The CG step penalty, it does have 3 CG steps. This one only has 2, because we see the penalty is 0.5. So we have 1 there, the other one's right there. And then again, because of the pyrimidine-rich bonus, so it does get a bonus for having more pyrimidines in the sequence. It gets penalized for having more purines. So the oligo length penalty is going to be 0 because we only asked for 20 formers, and that will give you a composite score. So I'm going to look at where our design went in our genome view. And within the genome view, sure enough, this probe wound up right here on that transcriptional repressor protein. So in this way, we were able to knock down that or modify that gene expression to exclude that protein translation. So we're going to close that and take any questions that you might have about Antisense Architect.
I'm going to scroll up. So it looks like one of the questions is, can this be best used for microarray probe designs? Could you use it in a microarray capacity? I imagine surface corrections or other.
So the microarray probe designs would actually, I think, be a specific application, and that would be something that we could build. Unless you were using the same messenger RNA library, if you were looking to knock down several proteins in the same target, then it could be used in multiplex. But it depends on what your focus is. So yes, if you're focusing on the same messenger RNA library, but if you're going over and above that, then it would be something that we would have to customize for you and build on your platform. So another question is, why does it penalize purines? And well, one of the more obvious answers to that is it doesn't necessarily penalize purines heavily, in that if you have a target that's purine rich, it'll still give you designs. But again, you're looking at some of the thermodynamic and kinetic issues of having runs of purines, particularly guanosines, that are going to form tetrads. And when they form tetrads like that, they're going to be form a unimolecular structure in the probe that is not going to have the energetic to form bimolecular binding, meaning that it's going to be very difficult and it's going to be a high energy cost to break that up. And also because guanosines in and of themselves tend to be promiscuous. So one of the main issues in antisense technologies is off-target effects. And so if you have a lot of guanosines in a row, or if it's built with a lot of GA, GAs, things like that, tandem repeats, then you could easily start to affect many of the off-target effects that we're trying to avoid. And so that's why, we sort of weight the C's and the T's as higher because they're more discriminatory when it comes to mismatches, and they're more discriminatory when it comes to actually matching. So it just helps when you're working toward trying to increase your specificity. Okay, are there any other questions for our webinar? Okay, the CG steps. So again, with the CG steps, what that does is it's pretty well known in the literature that whenever you have a CG, that depending upon the nucleotides that are immediately adjacent to that, they sometimes recruit antibodies. And so this goes then toward the immune response. And this has been an issue that's prevalent in antisense is, we can get the probes to their targets quite often, but then the probes will be sequestered or they're going to be degraded due to an immune response. And so we do look at the CG steps and try to avoid those. And the actual context is that if you have two purines that are immediately 5' to a CG, and you have two pyrimidines that are immediately 3' to a CG, then that 6 nucleotide signature sequence just recruits antibodies. So it can be an issue for an immune response, and that's something that we're trying to avoid. So that's something that the Antisense Architect also addresses. Okay, so we had, it was open. Okay, so any other questions? How do you price this?
Oh, so for pricing, that's something we can follow up on. I can follow up with you to answer any individual questions. We price it by the seat license, although in specific cases, if there's many users at a certain location, we have customers that use several licenses. We offer a site license with a volume discount. So basically by the seat to answer your question, but that's depending on how many users are at a site.
Okay, and I think that would also depend on if you needed a custom build, because quite often people are using different chemistries. They're using different backbone chemistries, sugar chemistries, and things that are specific to their proprietary databases. And we can take those and incorporate those into a specific build that you're able to use within your research. Okay, any more questions? Yes, yeah, and I just covered that. So, if you have a particular chemistry, you have a particular need. So the question was, can you customize this to my own needs? And yes, we can. So if there are even some heuristics that are germane to your particular target, we can input that on your platform, and we sell many of our targets as a platform. And so within that platform, we can move several capabilities in, several features in and out, and just customize it directly to your application. I know some people are a little weary of cloud applications, for instance. We can also have a custom application that's built onto your site using your own cluster. And so again, you can see pricing is difficult until we kind of parameterize your needs and what we need to do to bring that into your cluster.
All right. Well, if there's no further questions, I'd like to thank you for attending today's webinar. Again, I can be reached at Joe, J-O-E, at dnasoftware.com. Happy to answer any more questions or provide further reference materials for our work here at DNA Software. Norm can be reached at Norm, N-O-R-M, at DNA Software. Our telephone number is 734-222-9080. And to learn more about the kinds of problems we solve, you can find that information on our website at dnasoftware.com. So I'd like to conclude today's webinar, and thank you again for making time to attend it.
All right, thank you, everybody.